In this short video, I'll take you through the in-class resources we have for you for sectioning and staining plant material. These include preparing a wet mount, instructions on staining, and lastly of all, how to clean up after. There are quite a few resources in front of you when you sit down to your space in class. There's a staining bucket, there are some Petri dishes, there's forceps and probes, there's a sharps container, of course, safety being one of our number one concerns. There's some glycerol, some toluidine blue stain, there's razor blades, two sorts of razor blades for you. There's some blotting paper, there are a jar of slides, these are in ethanol, we reuse these. There are some cover slips, a wash bottle, a waste container for plant material, and we ask you to empty that into the larger bins in the labs after you finish. There's a box of prepared slides. There are some butter dishes for staining and sample prep, an eyedropper, a handy tool, as is your paintbrush. If you do any cutting, please do it on the chopping board, and a dish towel to dry your slides. The first step of preparing a wet mount is to get a slide out of your slide jar and to just dry it using your dishcloth. If you put some water in your butter dish using your wash bottle, you can use your paintbrush and apply a small amount of water to the slide. When we talk about a wet mount, what we're talking about is preparing your section or your material in water. For the first few sections that we do in the plant anatomy part of the course, we just ask you to remove a small amount of material. And when I say small, it's really a very, very small amount, and that can be placed in the water that you've already put on your slide. If you're not going to stain anymore, then potentially all you need to do now is to put a cover slip on, but in the next series of slides, I'll show you how to do some staining. Here I'm just going to stain the preparation that I just made before. So there is some material that's in a drop of water on that slide, and now I'm just going to place a drop of stain on top of that material. I'm going to use some blotting paper to blot up some of the excess stain. Usually you'd stain for about a minute. I'm accelerating this process just a little bit. And I usually use a torn edge of blotting paper because it is actually a little bit more absorbent. You can then use your paintbrush again just to pick up that stained material and pop it back into your butter dish with water just to give it a rinse. And after you've done that, you can clean down your slide, take that stained and rinsed material, pop it back on your slide with a little bit more water, and then gently place a cover slip on top of that material. You might need to squeak a little bit more water underneath your cover slip, and here I'm doing that just by using my paintbrush just to gently pick up small amounts of water and then just slip that underneath that cover slip so that there's the specimens fully covered in water. Remove any excess water from underneath your slide as well as on top of your slide before you put the slide on the microscope stage. What you don't want to do is to introduce any water to the microscope. This is particularly important because you don't want your slide to adhere to the microscope stage, and it will do if there's any water on the bottom of that slide. You can now examine the slide under the microscope. Note that the microscope setup notes are in your manual. To safely remove cover slips from your slide, you can do that directly into the sharps container as shown in the video. You can also remove any excess plant material that you haven't used to put that back into your plant waste and put that in the bin. The slide that you've just taken the cover slip off can just be put back into your container and can be reused next time.